want to introduce you to our speaker today, Berton Earnshaw. He is the Vice President of Recursion Pharmaceuticals, and he's going to talk to us about how to be a data science with or math degrees. So, Berton, thank you so much for being
Yeah.
here, and I'll leave you to it.
Okay, great. So thank you very much for the invitation to come and talk today. I'm currently in the offices of Recursion Pharmaceuticals, where I work. I'm currently a machine learning fellow here, so I do quite a bit of machine learning. But my background, I have a PhD in applied mathematics from the University of Utah, where I was part of the mathematical biology group there. I'm I'm noticing that the format here of Zoom is that I'm actually only seeing myself. I don't see any audience. I don't see anybody else on Zoom. I don't know if that is intended or not, but I don't know, Alvaro, if you can comment on that.
Yeah, it's because he has a webinar format. So all the attendees are in the background and you have solely the screen for yourself.
Okay, well, I suppose if people have questions, you're going to have to moderate that.
Sure. Yeah, we do have a Q&A bottom. So if you guys want to use that, or if you want me to pass any of the questions to Berton, let me know.
Yeah. So thanks. So I'm really happy to answer any questions along the way. What I wanted to do here was to tell you a little bit about what I am doing these days with my PhD in math. And yes, I'm doing data science. I'm doing machine learning in particular, a lot of deep learning. I just want to give you kind of some sense of that, and then be open or available for questions after that. So I'm going to share a screen. One moment while I get there. All right, let's see. All right, I'm hoping you can see that now.
Yep.
But as a machine learning fellow here at Recursion, one of my goals, my kind of overall objectives, is to make drug discovery better with the tools that come from math via data science and machine learning. And better can mean a lot of things. I'm going to talk to the way that we might be accelerating drug discovery through the use of a whole bunch of interesting data and tools like deep learning. So let's see. Yeah, these days, we became a public company last year, and these days I'm forced to give a disclaimer. So I apologize for the formality of this, but I'm asked to read this in its entirety. So I'm just going to say that this speaker, myself, as an employee of Recursion, and the views and opinions in this presentation are my own and not necessarily those of Recursion Pharmaceuticals. Their accuracy and reliability are not guaranteed. The material presented is general background information in summary form and may not be current or complete. This presentation is intended for educational purposes only. Full stop. Thanks for humoring me. Okay. So, drug discovery. So I think if I understand right, you're all undergraduates and you're thinking about, how you're going to use the skills and the abilities that you have and are acquiring through education and otherwise to have an impact on the world. And one of the great ways in which you might think of using these skills is in trying to solve one of the great problems of being human, and that is all the diseases that we are subject to. So some of these diseases are environmental in nature, but many of them are genetic. And you're seeing here a collage of people, all of whom are experiencing some type of disease, usually a rare genetic disease in this case. And rare genetic diseases are very interesting because even though they're rare, if you look at like kind of like the whole population of human beings suffering from one rare genetic disease or another, there are many of them, and recursion formed early on to kind of provide a way to overcome some of the challenges in drug discovery and pharmaceuticals generally, so that you might be able to treat rare genetic diseases. Because rare genetic diseases, they are rare. They have a small population of people who experience them, and uh, because of that, there's usually not a therapy on the market uh, available for these people suffering from these. And so, you want to be able to kind of have an impact on the world and make it a better place for patients suffering from these. And so, you want to look at like how pharmaceuticals are developed, and you want to say, is there a way that we can impact that? So let me tell you a little bit about what that looks like here. So what you're seeing here is a fairly well-known plot in pharma and drug discovery, where what you're seeing is on the x-axis, you're just seeing time, going all the way back to 1950. And what you're seeing is on the y-axis is the number of drugs per, this would be approved drugs, per billions of dollars in R&D spent. And what you're seeing is this downward trend. And this downward trend is exponential. You'll notice that on the y-axis, you're seeing this logarithmic scale. So this is really bad. What this is saying is that it's becoming exponentially more expensive to get an approved drug on the market over time. This is sometimes called Eroom's law, which is Moore's backwards. Moore's law was about the exponential increase in the number of transistors that you could put into a certain spatial a certain amount of space, and that should increase exponentially over time. This is an exponential decrease over time. So this is quite bad, and this is a real challenge for just the pharma industry generally. Is how what's causing this trend, and how can you overcome it? Well, Recursion, the company that I work for, and where I do data science and machine learning, formed about 8 years ago under the hypothesis that there's a number of new developments and technologies that have come together, recently, that should allow us to at least have a great chance at changing the trajectory here for Eroom's law. And they are the following: automation automation generally, but in particular in labs, like the ability to run a lot of experiments in an automatic fashion. Another convergence here is just the ability to collect and store lots and lots of data. This actually was a huge hindrance just a decade ago, but nowadays it's very cheap to collect and store data in the cloud, or even on-premise storage spaces. And of course, now you want to be able to use that data. So the really exciting developments in data science and machine learning, and in particular in deep learning, to be able to extract information from images. And so recursion really came about by saying, if we found a way that we could generate a lot of data about what's going on in cells when they're experiencing diseases and they have like drugs applied to them, could we use all of that to then improve our ability to do drug discovery? So so what you're seeing here in this new honeycomb slide are pictures of cells, and I'm going to kind of dig into this a little bit about what they are, or like what exactly you're looking at. But this is a form of data that's really quite new in terms of like how it can be used in drug discovery and developing drugs. And I'm just going to tell you a little bit about how we do that here at Recursion. But I just want you, I love this slide because these images are really beautiful. Really beautiful indeed. So let's just focus in on one of these. So what you're seeing here is a composite image of actually 6 different views of the cell put together in a kind of faux colored fashion so that we can visualize it in our typical kind of RGB dimensions. Um, and what you should notice here is that, or what I'd like you to notice here is just how much detail there is in this image of just these pictures of these cells. Now, what exactly are these? Well, let me break it down a little bit. So I said this was a composite image. It's really it's a composite of a bunch um, of individual images where what we did is we introduced fluorescent probes or fluorescent tags into the cell that bind to like specific parts of the cell. So here's one of those that binds to DNA. And this tag, this fluorescent tag, what it has is, well, what you can do is that if you shine a laser at a particular wavelength here on this tag, it will get excited and start to emit a light itself. And then you can image the intensity of that light. And this is the picture that you're getting here. So this probe or this tag is attaching itself to the DNA. And so what you're seeing here is like a picture of all the nuclei of all the cells in this image. And this is like one kind of image captured by a fluorescent microscope. Here's another view. So we introduced 6 of these. We sometimes call them stains. 6 of these stains or these fluorescent tags, and this one binds itself to endoplasmic reticulum. And so what you're now seeing is like the endoplasmic reticulum, the ER, in the cytoplasm surrounding the nucleus. So like still within the cell, but surrounding the nucleus. So you're seeing something about the cytoplasm. So you're getting something about, you're getting information about the extent of it, like the textures and things like that. And again, you're getting that per cell here. And then there are 4 other tags. So there's one for the mitochondria, giving you information about what the mitochondrial structures look like. There's another for Golgi apparatus and binding to the cell membrane. So you're getting some information about that. Another for RNA, and nucleoli. So like, you're getting some information about where the RNA in the cell is, where it is in the cell, and like how much of it, by the intensity signal. So again, you're seeing more structure of the cell. And then there's also another, which is actually not necessarily the cell, but it's like surrounding structures called actin fibers that kind of tell you about just how the cells are being supported. And so we bring these 6 images of these cells together into one 6-channel image. And again, we're kind of coloring it in a particular way so that you can kind of get this overall view. You can kind of see that like the actin is this red color, the nuclei are this like purple color, and so forth. And again, what I want you to just notice here is just how much detail. I'm kind of zooming in here a bit. You can see this kind of rich structure here, and I can zoom in even more, and you're seeing the nuclei and the nucleoli, and you're seeing the cytoplasm, both the mitochondria and the endoplasmic reticulum, the Golgi apparatus, and the actin fibers and so forth. So like you're just getting a lot of information, okay? And so the idea here is that this is a kind of data about cells that isn't typically used in pharma. However, it's really interesting because there's a lot of detail, and you can produce a lot of it at basically like very low cost once you have bought all your microscopes. Like basically running these experiments to collect an image like this is really just a few pennies per image once it's been done. And right now, I mentioned that Recursion, where I work, has been around for about 8 years. We've gotten to the point where we produce well over 1 million of those kinds of images every week, meaning like roughly we're running about 1 million experiments a week. Well, actually right now about 1.5 million experiments every week. Um, that produce like whose readout um, is this image. And and so, like what's that for? Well, what you want to be able to do when doing in drug discovery is to be able to say, like, you want to be able to model a disease, and then you want to be able to see the effects of the of like different therapies on that disease. So in this case, we're gonna talk about drugs, in particular small molecules. Um, and so what you want to do is you want to be able to model the disease, put the drug on these disease cells, and then look at those cells after the drug's been applied and say, like what did anything good happen there? And so like the question of how to answer that question, I'm going to go through in a little bit here, but I want to just I want to kind of give you that idea about that like here's where like math and modeling are starting to come in, is that we need to do a couple things. We need to be able to take these images and turn them into something a bit more useful, a bit more amenable to like mathematical, statistical, machine learning approaches. And we want to be able to do this on a very large scale. Because what we want to be able to do downstream is answer these kinds of questions of like, did a drug improve, the state of cells, like the cells that are in a disease state? What you're seeing here is kind of a picture, maybe a little bit hard to kind of understand. There's a lot going on here, but what you can think about our platform being able to do is take various types of human cells, or cells in different kinds of contexts. They could be they could be excited or inflamed or whatever in certain ways, that have them model diseases, and then run through our platform, be turned into some basically like list of features that then where we make all these decisions. That's what's over here on the right. Things like being able to work out kind of how different like different drugs at different concentrations might affect a disease. We produce a lot of this data. I was saying we do about 1.5 million of these per week. We really, at this point, we have well over 100 million images that are kind of like the ones I've been showing you, that represent different cell types under different kinds of genetic or chemical perturbations. And what's really cool is that at some point we decided that we would start to make some of this data public and available to whoever was interested in it. And we have a website, rxrx.ai, that you are welcome to go look at and explore. You'll find a number of data sets there. Right now there are 4 data sets that, and here are the characteristics of those data sets that that are available to you, that are under very open licenses. In fact, when COVID-19 hit, I'm going to kind of switch gears here and talk a little bit about COVID-19 in a second, and some of the studies we did around that. We produced a bunch of data, and we made that available, and it's even available under a commercial license, meaning people who are free to use it to develop drugs commercially from it if they found it useful. But anyways, just wanted to make you aware that this is there, and if like you find some interest in what I'm telling you today, there's a place to go and download data, and you can start to kind of play around with it. We also hosted a Kaggle competition. I don't know if you know what Kaggle is, but Kaggle is a place where that hosts machine learning competitions, and we used the first data set here, RxRx1, as the data set for that competition, and we set a task for that, and it was a really exciting time. Just watching the progress on that particular competition was really fun to see what happened there. And then also we were able to use some of the ideas that were generated there for our own in-house work, and we actually were able to hire at least one person from that competition, the winner of that competition, which was also exciting. But I mentioned a couple times this idea of needing to turn these images into some sort of useful representation for drug discovery. And the way that we do that, at least the amount that I can tell you at this point, is that we use deep learning, or we use neural networks to basically perform a bunch of tasks where the data is the input to those tasks. So what I mean by this is that there's like these tasks will be sometimes very simple tasks like classifying like what we did to the cells, like what perturbation did we observe, or sorry, what intervention did we introduce to the cells, and like we know what we did to them, so we have that label, so we can ask this neural network to figure out like how to do that very accurately. We can also ask it to do more complicated things like learn relationships between genes, like some genes, sorry, between perturbations, I should say. So like so you may know that genes are like the code for proteins, and proteins are the basic building block of like everything in your body, and what's cool about proteins is they perform a lot of important functions, basically like all the functions in your body, or at least like groups of these like put together in various different ways. And so because they work together, there's a bunch of relationships that one might expect, sometimes called gene-gene relationships. And so, like for instance, two proteins might come together and form a complex that is then part of like a signaling pathway. And so we expect there to be a really strong relationship between the genes associated with those proteins, because their proteins work together in a complex. And we can basically like, what we can do is we can do something like the following. The perturbation to the image might be we knock out that gene, meaning that the protein should not be in it, like exist within the cell. And if we do that, we take pictures of the, we collect these images of these cells, we will expect to see the cells look a certain way. If we go and knock out the other gene in that complex, we would expect them to look in the same, like in that same way, because in both cases that protein complex is being prevented from working because one of its components is missing. So there's like relationships like that we can teach these networks to learn as well. And there's a whole bunch of others that you can think up and get the network to learn to do. And what's really cool about like neural networks and deep learning in general is that because this is a like a stacking of a whole bunch of like just simple transformations, linear transformations followed by simple nonlinearities. You have at every stage in that, right? We call them a layer in neural networks, but at the end of every one layer, we have basically a representation of the original input, just kind of transformed that layer. And like the closer we get to these tasks in that whole neural network, the more information that's coded in that representation of that layer for doing those tasks. And those become really, really powerful representations of the original input, right? So we could go from a really high dimensional image, right? So like our images, when they come off of the microscope, the size is 2048 by 2048. That's the like the resolution, and then there's 6 channels. Well, we're going to compress all that down into, say, like a much smaller number of features, and then use that compressed representation to do all of our downstream drug discovery tasks. And so the more information we kind of can extract usefully by providing all these tasks, like the better off we are in terms of being able to do downstream tasks with them. So how do we use these things? This is sometimes called phenotypic drug discovery. I'm just going to give you kind of the real quick explanation, and then I think I'll stop and take a bunch of questions. But the basic question you're trying to answer in drug discovery is like, can we make disease things better? Can we make them healthy? And of course, what we want to do is for humans, but we can't just experiment on humans. We do experiment on humans. This is called human clinical trials, but you only do that once you have a drug that you're very confident in is going to be effective for against the disease, and also is going to be safe for humans. We can't just go, obviously, for all the for all the obvious reasons, you can't just go trying all new drugs on humans. So we have to do these things in models like these models of these cells that I've been showing you. And so to do phenotypic drug discovery means that like we have phenotype means like the way things look. So genotype is like what's our genome like how are genes being expressed, and then the phenotype is like what's the effect that we can observe from that particular genotype. So it's like the way that the cells look, the morphology of the cells. And so disease might look one way, healthy might look another way. And what we're looking for are drugs where, like, at a particular concentration, we've really taken the way that the disease cells look, and the effect of the drug is to make them look far more like what the healthy drugs look like. So like how do you think about that? Well, this is definitely where math comes in. So I talked a little bit about how neural networks can give us a representation. It's just a vector in a vector space, right? Let's use the number 128. It might be a 128 dimensional vector space, and each one of our images is going to be like put into that vector space. We sometimes call that embedded in the vector space. Okay, so we have these vectors, and so we have this like all the vectors associated with, say, like the disease. That forms some sort of distribution in our space. And then we have all the vectors associated with healthy, and that forms some sort of distribution in our space. And as we apply a drug and increase its concentration, what we're hoping to see is that we're going to see the vectors associated with those images moving from out of the disease distribution and into the healthy distribution. And there's a lot of ways in which you might measure that. But in essence, they all rely on kind of the underlying geometry of the vector space, and I'm showing you like some picture to kind of give you some idea here. One way we might approach this is to have our disease so we have our two distributions, our disease and our healthy. And we can sort of draw a line between like the means or some other way of central tendency. We can draw a line between that, and that kind of defines like an affine subspace in our vector space. And what we can do for any other vectors, we can project onto that vector, or onto that subspace. We can project that vector onto there, onto it, and then we can think about its complement, its orthogonal complement as well. And in doing so, what we do is we get a kind of notion of like what part of this vector is on target, like that's associated with a disease, and what part of this vector is associated with just like other effects in the space that aren't necessarily the disease. And in this way, we get kind of two scores that that tell us about like effect on disease, and then like keeping track of other things. And a way you can kind of visualize this is this plot right here, where we're projecting basically all these vectors into this 2D space, where you have the x-axis here is like literally like the projection onto this subspace, and then the off disease part here, the y-axis, is just like the magnitude of what's sometimes called the rejection, or just like the orthogonal complement for that vector. And then what that's telling you is just like what's happening, just not what's happening in this vector, just not in this particular direction. And again, like we call it off disease because you can often think of it as like off target. Like what else is this like compound doing that's not disease related? Because when doing drug discovery, you want to be able to find an effective drug against your disease, but you also want to make sure that's not going to like affect other parts of the human that could be like toxic, for instance. So you can kind of draw these things in two dimensions. You can project your distributions, and then you can look at these trajectories. Each one of these is like um, is the same drug. Each one of these um, like piecewise linear paths here is just the same drug at different concentrations. And what you can see is that some of these drugs, um, are actually go from this disease distribution and move us towards the healthy distribution. And some of these things start to do that and go off in other directions. And some of these drugs don't even leave the disease distribution. And so in using tools like this, you can start to pick out which of your drugs is looking better. And remember what I told you is that we have the capability of running, like over 1 million experiments a week. And so you can imagine running a lot of drugs against any particular disease. And in fact, we do a ton of this. I'll just skip here. We do a ton of this all the time for different diseases. And when the pandemic started and we started, to understand about the effects of, or we started to, experience the effects of there being a very contagious virus spreading, a lot of groups, obviously pharmaceutical companies, but a lot of academic groups as well started to look into how they might help in terms of either preventing infection or dealing with the infection after it's happened. So at Recursion, we really quickly put together the ability to run our experiments, the ones I showed you, like basically where we do the imaging readout, and see if we could pick out which drugs appear to be effective or not against like different forms of like disease associated with the SARS-CoV-2 infection. So like one is like the early stages of the infection. This is like literally like dealing with the way that the virus changes cells, and the other is the response to all that infection. So a lot of times when people die from COVID-19, that they don't die from the infection necessarily. They die from the way that it causes like your immune system to kind of respond kind of out of control, something called a cytokine storm. But and this is why it's linked to like to other comorbidities, right? Like if you're already in a state where you're fairly sick, like this is going to make things far worse. But anyways, we did this and we made a bunch of predictions. We put this out in like a really quick preprint that's out there for the world to see, where we made predictions about like certain drugs and whether we thought that they would be effective or not. When I say predictions, I just mean like we're basically reporting the outcome from our assay. Obviously, since that time, there's been a lot of study of the effects of COVID-19, and there's been a lot of randomized clinical trials that have been able to say, are certain drugs, effective against either the early or late stage of COVID, and so what we were able to do is we were able to look at the outcomes of those, look for the drugs that we actually tested when we did our study, and see like does that match with what we predicted from our outcome. And so far there's been 7 of these clinical trials that have finished for drugs that we had in our study, and for 6 of those we actually predicted it correctly. I got them listed here in this table, where just doing the kind of representation of those images, and then kind of doing the similarity comparisons in this high dimensional vector space, we're able to say that like hydrochloroquine and a couple of these other drugs were not going to be effective against COVID-19 as was shown in the randomized clinical trials, and that drugs like remdesivir were effective, and again corresponding with the outcomes of the trials. And in the one that we did not predict correctly, going back and looking at how we modeled the disease and how this particular drug works, like it actually makes sense that we weren't able to do it because we weren't actually capturing the mechanism by which this drug um, uh, um, actually like its effect on cells or like on physiology. So we made the wrong prediction there, but we understand why, and that's important as kind of error analysis in terms of how to make future predictions better. Anyways, that's I just want to tell you that story because, like, in a nutshell, this really exemplifies what like the power of what we do at Recursion. And again, like bringing together all these different technologies, lab automation, the ability to store uh, produce and store lots of data, and then to analyze that at in a very high throughput way with tools like machine learning, to be able to speed up drug discovery. In the sense, in some sense, so we did this in a 4-week span. Like we went from like the conception of the idea of doing this to producing the data, and we had these predictions, which in 6 out of 7 uh, 6 out of excuse me 6 out of the 7 trials we actually predicted the correct outcome. So like it's a small proof point, but it shows how powerful tools like this can be in terms of being able to pick out drugs very early and then quickly getting them into clinical trials so that the FDA can then approve them for use for treating the various diseases. So yeah, so I guess I'll stop there, and I'll leave this slide up for a second as we take questions. Just to point out that um, if you're at all interested, in these kinds of things, it'd be great to hear from you. There's the email here that you can contact, or you can contact us generally at. There's the website arcs.ai, and of course we have a careers page if this is something that if you're at that stage, you're looking for jobs. We'd love to have you apply. So thanks. I'm happy to take questions at this point.
Right. Thank you very much, Berton, for the presentation. Really, really interesting. So I want to start the session question with a question of mine. So you were working on vectors and high dimensional vector spaces. So how high dimensional is that vector space?
So I was throwing out the number 128. So like that's a typical size. We often can be in a vector space much larger, where it's over 1,000 dimensions. And if this can be quite challenging actually to do like statistics in these high dimensional spaces. If any of you have taken courses in statistics, learned about like hypothesis testing, you'll probably learn something about the power of your test. And this power unfortunately goes way down as the number of dimensions goes up. It's one of these like aspects of something called the curse of dimensionality. And it just has to do with the fact that like your data just doesn't cluster together as well as the more dimensions there are. So like there are trade-offs to like high dimensionality giving you a lot of like information, but then being able to use that information to do like significance testing. So dimension of 128 is kind of a typical one for us because it kind of balances between those two things, but it's really hard to say like that's the right answer. All I'll say is that like it's always a power of 2 for some reason. We just always use powers of 2 or sums of powers of 2. That's maybe the only other constraint, but yeah. That's a typical size.
And I imagine you can get characteristics from the different orthogonal spaces and even maybe from each of those 128 spaces, right?
Yeah, sorry, for some reason I couldn't hear that very well. What about the 128 spaces?
I imagine that you can get characteristics and information from each of the orthogonal spaces in the 100.
Oh, yeah, absolutely. Yeah.
All right, so I have some questions in the chat. Let me check. So Alexa asked, what is something that you have worked personally with this data subject?
Yeah, so I don't know if personally meant like if I'm doing like a personal project that's not related to work, or with this kind of data. I've done a lot of work with this data. I'll just say like from just as part of my role as an employee here at Recursion.
I think
you know,
she
like
was asking like for something more specific, right? Like,
Well, no, yeah, not personally, like kind of like what your contribution has been.
oh yeah. Yeah, great. That's a great question. Yeah, so like the main models that we use for um, doing the representation of the images, is doing the representation learning, where we turn the images into vectors of numbers via training neural networks. Like that's my project, and that's been my project basically since I've been here. I've been at Recursion for about 5 years, and just working on like how do you improve that? How do you know that you've improved that? How do you evaluate that appropriately? Um, what I'm not showing you is like other ways that we use machine learning at Recursion. So other ways is like there's a lot of really powerful machinery, again using neural networks, that allows you to learn to, like, given a set of conditions, to literally generate a molecular structure for you that you've hoped, that your network's powerful enough to have like the right properties to be like a good starting point for a particular disease. So like the idea is that you can give this network information about your disease. Like you can condition this network on your disease, and then it's basically a sampling. You can think of it as like having learned to sample from a distribution, and it samples from the areas of highest probability. And so, being able to sample, to generate molecules, or sample from this distribution that produces a molecule, is another area of work that we're currently working on. That's really powerful and kind of goes deeper, on neural networks, deep or in the deep learning area, and really, like if we can get it to work well enough, really cuts down on time, because it's instead of like having to do all these like very large screens, or then do all these kind of secondary assays to validate, you can just go straight into validation from the generated molecule. I don't know. There's a couple of examples of the things that I've worked directly on here.
hey, there's a couple more of questions.
Yeah, I've actually opened up the Q&A tab here. So
Okay, great. That's great.
yeah, so then I see the next one is, what are the skills a graduate we need to get in research of the level Recursion does? Yeah. So I mean, so I'm going to assume that you're all mathematicians, and that you're interested in things like data science and machine learning. So in general, what we look for and what we hope to, for candidates to have is really a good background in being able to solve problems using math, using algorithms, using code, not necessarily like that you're writing a website or something like that, but you can use code in sort of a scientific pursuit and be able to kind of solve problems, in code. So, just like strong fundamentals in mathematics and statistics and probability, and then being able to code up ideas and, try them out, is a really important, foundation. And then, just demonstrating the ability to do that. So a lot of times, we if you're just graduating, you may not have had much of an opportunity to really demonstrate that out of school. But this is what we really look for, people who have demonstrated that outside of like just like the classroom setting. So, taking internships in areas like this, where that give you an opportunity to learn how to do this in like in some kind of like industrial setting, like at a company like Recursion, is really valuable, because it really demonstrates to us, that kind of ability so data science and machine learning is like a humongous set of topics, really, right? It's not just like, oh, you do data science, so like you do this, or you do machine learning, you do this. There's like a whole bunch of these things. So obviously, like having familiarity with like, across the board with a lot of different tools and tool sets is really useful, because that kind of broad understanding allows you to have just a wider range of tools to work with problems. But it's not like that's absolutely necessary, although that's useful. But again, like demonstration of the ability to use at least some of those tools to solve a problem outside of school is just really, really, really important. And we have various levels of positions. So there's entry level, like analyst or associate data science positions, all the way up through like very senior, machine learning research roles, right? So like within that spectrum, there's a lot of different opportunities, and early on in your career, you're hoping to kind of get in to one of those early roles, and then start to grow there through your experiences, through the work that you do. So I could answer more specific questions on that if someone wanted to ask a more specific question, but hopefully I gave you some kind of understanding of both like the kind of foundational skills and also the how to demonstrate that you know how to use those skills. The next question here says, what are the most important classes you have taken for the job you now have at Recruit, or you have now at Recruit? This is a really great, it's a really great question. So, I did a PhD in applied math, as I said, and I was essentially doing dynamical systems, right? Like I was solving, I was designing and solving differential equations, systems of differential equations. I don't use any of that now. I don't do any differential equations. And this is a really interesting aspect of how like the market works right now for people with like mathematical and statistical backgrounds, right? It's there there are definitely opportunities in many, like to use differential equations. Like you could, I don't know, you could go become an engineer or a physicist or something like that, and I'm sure that there's lots of differential equations being solved. However, many of the jobs available to you will be more in this kind of data science and machine learning realm, and they really rely very little on differential equations. It's a lot more of like statistical modeling, and machine learning is just kind of one of the tools there, where you're learning associations between different sets of data, right? Like your input and your outputs, and so like the tool set there is just quite different. The tool set there is really about figuring out ways of using effectively the data that you have to learn these relationships, so that you can use them in future settings, right? So like we again, just kind of like bring in an example at Recursion, one of the most important things we do is to represent the images in a way that can be used in downstream like analytical studies. So being able to do that is about being able to train a neural network to learn to like associate the input, the images, with all these different outputs, like information about how, like what we did to the cells that are in the image, and its relationships to other like biological or chemical entities and so on, right? Like, and it like that's not that's it's math, and it's like, and it's statistics, but it's not exactly what you're doing like in your classes probably when you're getting a math degree. However, like very much understanding how all that works, and like what changes you would make to like say the training procedures, it is very much guided by good sound like mathematical understanding and statistical thinking, right? Like we are taking derivatives all over the place, very complicated derivatives when we're like training a neural network. Being able to like reason about derivatives and gradients and how like chains of those work together is actually a big part of being able to think about like how a neural network works, and like if you're looking to improve the performance, like where you would maybe make the improvements. And so yeah, so coming back to the original question, I never took a machine learning course, so, and I mostly just do machine learning now, so I didn't actually take a machine learning course. So, but the most important classes were really ones about being able to do rigorous, thinking about like, solving problems, mathematically. So like, a lot of like the courses in analysis and linear algebra, in numerical analysis and optimization. And I say like I don't use differential equations, but like a lot of the thinking and the dynamical thinking there is very, very useful as well. So I don't know there's one like really most important course, but like what you get out of it, like an applied math degree, for instance, is really helpful in the kinds of things that I do. And of course, like some of the statistical and the probability statistics ideas as well. Yeah. I feel weird because I'm not like seeing anybody other than Alvaro, so I don't know if like I can't tell if like questions are being answered appropriately or whatever. But there's one more question here that says, what programming language does Recursion use? Our math major has us use Python and R mostly, but there are some classes that introduce Mathematica and LaTeX. Great. So yeah, so we mostly use Python. There's some very limited use of R, and you use R only when you have to. At least that's for us. And when you have to, it's like there are some statistical tests that are only coded in R and they're not in Python. They're not like in some Python package. And R is really amazing this way. Is really useful this way. But in general, we use Python mainly because it interacts so well with the rest of like the like analytical environment that we're in here. So like Python, for some reason, become like the standard API for a lot of other things. For instance, like deep learning, there's all of the deep learning frameworks have a Python API, and that's like the primary one that's used. And then you have all these other general libraries and also bindings to like other languages, and so it becomes like a really useful general purpose language to be able to do a lot of different things. And there's Python has a lot of limitations, but what it does have going for it is just like ease of learning, ease of use, and not really getting in the way of itself. To be able to be this API for the other things. So Python definitely is the most important and used. R is I would just say like it's not like you have to learn tons of R. You just need to know that like if you have a weird statistical test that you're trying to do and you can't find it in your Python libraries, you almost certainly will find it in R. And so you should go there and figure it out. And then like I mean nobody uses Mathematica. I guess just because we're not doing anything like I don't know. We're just not doing like symbolic kinds of stuff, and I don't know. Like Mathematica is quite broad, so we're just not using it as a tool. Like we just don't need it. But we use LaTeX for writing papers, for instance, right? Like LaTeX is the standard mathematical typesetting API, so that's what we use it for. So like designing posters and papers, and anytime we need equations, you basically got to turn to LaTeX. If you want it to look nice, you can use like Google Docs or something like that, but it won't look as nice. So yeah, good question.
Is there any other questions you want for our speaker? All right, Berton, thank you so much. Dr.
Yeah.
Earnshaw, sorry about that.
Yeah, that was great.
Thank you for being here. Let's thank our speaker.
Thank you very much. I appreciate the invite again, and good luck with things. And if anybody wants to contact me subsequent here and ask some additional questions, that's fine. I guess I should have had my email address on this last page, but Alvaro can share it. But it's just my first name, Berton.Earnshaw, my last name @recursion.com.
All right, Dr. Earnshaw, thank you so much for being here with us, and have a good day.
Yeah.
We'll be in touch.
Thank you. You too. Bye-bye.